I'd like to welcome you all and thank you for coming to our kickoff community lecture series this evening. I'm going to start out just by giving a sort of very quick overview about the center, who we are, what we do, why we're here, how we came to be here. And then Dr. Brian Cummings is going to talk to you about traumatic brain injury, and which is one of the kinds of basic research, basic science models that we do here in the building. And then Dr. Jefferson Chen is going to take it over and talk about the clinical trial that has been ongoing at UCI using mesenchymal stem cells as one potential novel therapy for that, to take you through kind of the whole bench to bedside process, and I'll come back to that in a minute. And our goal here really is to do some community outreach, to engage with you all, to try and get new people to come into the center, learn about science, get interested and excited and curious, and think about what stem cells mean to us and where we've gotten with stem cells in California over the last 10 or 13 years as the California Institute for Regenerative Medicine has really been working hard to do these transformative kind of studies and break some new ground for clinical and basic science research. So this is a classic cartoon for stem cells. You can be whatever you want to be when you grow up. And it's good advice for kids, but it's also really true for stem cells. And I'm going to tell you why here and just introduce very briefly the idea of stem cells and how stem cells work. The basic concept is that these are cells that are, we now know, in every organ, every tissue that you have in your body. And they have the capacity to do one of two things. They continue to divide, and they either enter a path that makes them differentiated, so they become a part of a tissue, like an organ, your liver, your kidney, your brain, and so on, or they stay in kind of a pool that can make a new supply of cells if there's damage or disease or we need to have cell replacement for some reason. And I like to use this diagram to illustrate stem cells because it tells the whole thing kind of in one simple cartoon. The idea is that a totipotent cell, a cell that's present right at the very beginning of development and can make any cell in your body, is represented at the base of this tree. But as you go farther up towards the branches, these cells do exactly what number one is in terms of the path. They become more and more specialized, so that as we go up this branch here, which is where I work, and you get to the nervous system, you have cells that are still stem cells, but now they can only make the cells that belong in the brain or the spinal cord, neurons, oligodendrocytes, astrocytes, for example. And as you go even farther out this branch, now you get a cell that's terminally differentiated. It's finished, it's done, it's all grown up. And now it's one of those cell types. It's a neuron or an oligodendrocyte or an astrocyte, and that's how it's contributing to the function of that organ. So here at Stem Cell Research Center, we study stem cells of all kind of shapes and kinds, from the very, very beginnings of stem cells, those ones that can make anything, on up through how we can control the differentiation of these cells and use them potentially therapeutically, studying those in clinical trials. Stem Cell Center got off the ground in 2006. Actually, our first director is here this evening, Dr. Peter Donovan, who really did a tremendous job of taking us from something that was non-existent, no structure, no building, no organization, no faculty, to what we are today, which is 23 faculty that have working laboratories studying all different aspects of stem cells that live in this building, and actually 48 faculty that are distributed across the campus that we collaborate with and engage with on a day-to-day basis. Most folks here are in the School of Medicine, but we have a really strong representation from biological sciences. That makes sense. And maybe surprising for some of you other places like pharmaceutical sciences and engineering and the School of Law and the School of the Arts, because what we engage in is really very cross-disciplinary. This facility is about 100,000 square feet. We have a mix of lab space, office space, clinic space, and for example, the conference center that you're sitting in right now. We also have a number of core facilities that are really, really important to helping people do their work. And these range from specialized equipment that anybody on the campus can use, on up through some specialized support mechanisms where we train people actually from all over California in how to do stem cell culture, send them back to their programs, and help them get on their way with their own stem cell projects. And this includes training for graduates, for undergraduate students, community educational events like the one that you're in right now, and of course scientific symposia. We welcome you to come for any of those things in the center, and there's actually some information that's at the back of the room over some of the events that we're having over the next year here. Funding-wise, we have a very active center in California. So California Institute for Regenerative Medicine has been absolutely pivotal towards jumping stem cell research forward, and investigators in this building have over $100 million in grants that have come in from those sources. But we also get money for the basic science and the clinical work we do from the National Institutes of Health, for example, and the National Science Foundation. And of course, philanthropy is very, very important to us, and I'll come back to that in a few minutes. We have a mix of things that we do here, from basic science research, so systems and cells and molecular biology, techniques and tools that we can apply to stem cells and how to make stem cells work better. Biomedical engineering. In fact, we're kicking off a new tissue engineering program. We just got allocated by the provost's office a set of new faculty positions in order to build in this area in collaboration with both the main campus, biological sciences, and with the School of Engineering across from the School of Medicine. And we actually do quite a lot of work in terms of ethics and policy, and we've hosted a number of conferences that are really getting at the core of what does it mean to use stem cells? How should we be thinking about those? What's the responsible way to go forward with this as the field is moving just so quickly? What you're going to hear tonight a lot about, though, is translational research. And these represent some of the very different areas in the building where people are working at this translational borderline. And our community lecture series that's going to be ongoing over the next year is really built around educating people and doing some outreach to the public in all of these different areas. And again, the back of the room is some information about those. All that information is on our website. And we hope to see you again at some of these other lectures. I gave this introductory little bit to the UCI Retirees Foundation about a week or so ago, and someone asked the very clever question, what is it that you mean by translational research? And I said, I should put a slide or something in about that. Because we use that term all the time, and it's something that I realize now not everybody is engaged in the same way, and so I thought I'd spend a minute there. The kind of facile way that we talk about it is being bench to bedside. That didn't help explain it a whole lot, so I'm going to spend a minute on this. What we mean by that is most of the research that we do in this building is kind of up on this hilltop. We're trying to understand how these cells work, how we can make them work better, what sorts of molecules signal, with very foundations of basic science biology for how things happen and what the mechanisms are. But really our long-term goal is to get over here to this mountain, and that is to think about treating clinical disease. And it's a pretty big chasm. And from that, what a lot of people don't think about, that's what we call bedside, is clinical practice. And that's a whole nother hurdle with its hazards. And by that, what we mean is changing how clinical medicine is done on a day-to-day basis, not just at an academic medical center like UCI, but in your primary practice office, in how people get referred and go out and get treatments for cancer, for diabetes, and so on. And so this is a more classic image of that. What I want to point out here is in this clinical bit, as we move on towards practice and approved drugs, is what is commonly called the valley of death. And so I thought I would spend a minute, again, just explaining that. Here is where we work mostly in this building, right? On the very discovery part, basic science. This slide is built around the idea of developing small molecules, but stem cells have a lot of commonalities there. The basic principle is that you have to do a lot of screening to get down to a small number of things that go to clinical trial, to get down to something that passes clinical trial and gets across the mark into actual approval or into clinical practice. And so most of the work that happens behind everything that we do in medicine today is on that very bench side, on the discovery science side. And that's really the theme that I want to convey. Without discovery science, we don't have a pipeline for anything that comes after. So where does CIRM and translational research fit together? Well, translational research seems really slow. It seems slow. My main work is in spinal cord injury. When I talk to folks that have spinal cord injury, or of course their families all the time, it feels like things take forever. But in reality, while the caption of this slide, I'm not sure everybody can read it, it says, you don't have anything that another 75 years and some stem cells can't cure, right? 75 years feels like forever. But stem cells have really moved, and translational research with stem cells have moved at a rocket's pace over the last few years. In fact, there are 49 California Institute for Medicine funded clinical trials that are currently ongoing here and based around the country in reality that derive from Prop 71 and supporting stem cell research, the decision that all Californians made to support stem cell research and try and drive this field forward. And in terms of timing, the of the clinical trials that I've been involved in, where myself and Dr. Cummings have done preclinical work to move those forward. In fact, we started doing stem cell research in our labs in about 2002. And the first clinical trial for spinal cord injury, the first in-man clinical trial with stem cells for spinal cord injury from our work, was launched in 2010. That's only 8 years. So when I say these types of approaches are moving at kind of a rocket pace, they really are. And we're all sort of scrabbling to keep the pipeline in place to support that. So our clinical research profile here is actually pretty wide. Everything from a special set of rooms that we have on the first floor to develop outcome measures, how do we know that we made somebody better or we didn't, to preclinical development of cells that were used really in the first 2 clinical trials that went forward. First in-man trials for spinal cord injury was all done by investigators in this building. Same for retinitis pigmentosa as an application. And what I want to spend just a minute and tell you about now is the UCLA, UCI, CIRM, California Institute for Regenerative Medicine, Alpha Stem Cell Clinic, ASCC network. Because some of this community lecture series is possible by the fact that we have these clinics as a foundational change for how we can go forward with stem cell research in California to enable supporting infrastructure to do responsible, well-informed clinical trials that will really tell us what's working and what isn't, and what's the best way to move things into practice. So UCI and UCLA were part of the network of the first 3 sites that were funded by CIRM in 2014. We just added 2 sites to the Alpha Stem Cell Clinic network this last year at UC Davis and UCSF. And this has turned out to be a really great cooperative network. I don't expect you to be able to read this. If you want information on ongoing clinical trials on our pipeline, here's the website right here. But I just want to give you a sense of how active this has become, again, reflecting that 49 trials that are ongoing with CIRM right now. So I told you that most of the work that we have to think about doing is on this bench side of the equation, right? We have to screen a lot of things. We have to understand a lot about the basic biology before we can move forward. And the way that we have tried to do that here at the Stem Cell Research Center is in a program that was kicked off by our last director, Dr. Sid Golub, and that's a seed grant program, which has turned out for us to be enormously successful. We've been able to raise and distribute about $350,000 to get people off the ground for new ideas. But that $350,000 has brought over $6 million in new funding. So this is funding from the federal level, National Institutes of Health, from California Institute of Regenerative Medicine. And what we're very proud to report is that that is a 17.5-fold return on investment for how that's gone so far. So we're pretty excited about our capacity to support seed funding. Why is that important? So you might think this is kind of a funny thing to kick off a community lecture with, but I think it's really important for everyone in the community to understand what the face of funding for science looks like nowadays. So this is a graph from the American Association for Advancement of Science, and it shows where funding going back to the early 60s to present day has come to in the United States. And whether you look at funding for science either as a share of the federal budget or as a share of GDP, we're sitting at about 25%, about a quarter of the level of where we were when we were trying to put the first man on the moon. So our investment in science as a nation has really declined over time, and it's eroded our capacity to feed that pipeline. And one reason I want to point that out to you is that we as Californians made this tremendous investment with Prop 71 and in the California Institute for Regenerative Medicine, and it has changed the face in timeline of what we can accomplish with stem cells. So I hope that you can all sit here tonight and be a little bit proud about that and think about where we've gotten as a state with that investment. So thanks for coming. With that, I'd like to introduce Dr. Brian Cummings, and we need a moment to change over all these multiple microphones. So Brian is a professor here. I'm just going to shout right now. Brian here is professor at UCI in the departments of physical medicine and rehabilitation and anatomy and neurology, and of course in the Stem Cell Center. And what he's going to do is take about 20 minutes or so tonight to tell you about his research with a different kind of traumatic injury from spinal cord injury, traumatic brain injury.
Okay, can everybody hear me? All right, so first off, thank you for coming. It's now moving on to the main event. And really, these slides are going to be posted on UCI's Stem Cell Center website, so you don't need to take notes. You're welcome to take pictures, you don't need to. We're going to post it all, including the video from tonight, and the question and answer session. So I like to start off with my acknowledgement slide, because I'm the one who gets to stand up in front of the public and say, here's all the great stuff that I've done, which is a complete lie. This is the stuff that a team of people have done, a large team, and if anyone's very observant, they will notice the director here in the tree, because truth be told, we're actually married too. So that's the background secret. The other part is I am chairing the Public Outreach Committee, and our goal is to have you come back as many times as possible and learn more about what we do here. You're welcome to invite friends, and we'd like to grow this so we can educate our local community. So I want to thank everybody. It's a team effort. I'll thank the TBI team a little bit later, but I just want you to realize it's a very big team effort. And also, it's a team effort to host these presentations. So I want to thank Andrea Ortiz and Chris Nishi, Jackie Tidball, Amber Harness, and Janice Briggs, who have helped behind the scenes to organize all this. So thank you, everybody. I'm going to give you a very outline. What am I talking about? Well, I'm going to go really simple down the line here and come back to this outline so that you know where you are. I'll give you a background on traumatic brain injury. What is it? Then I'll talk about work in my laboratory on neural stem cells, repeat mild concussions, and using stem cells in a dish to model brain injury. And then after that, I'm going to give you a little bit of background about SanBio's mesenchymal stem cells that are in their clinical trial. And then Dr. Chen is going to come up and tell you about how we put those cells into patients. So, the numerical toll of TBI. Really, the reason why I switched from spinal cord to TBI is because TBI is really a silent epidemic. It's the #1 killer of children and young adults in America. So it's a massive public health problem. TBI affects 1.7 million Americans each year. That's 3.8 million if you include people with concussions. And this is more than the people with brain, breast, colon, and prostate and lung cancer combined. So we've got a pink ribbon for breast cancer. We don't have a ribbon for traumatic brain injury. It really is a silent epidemic. 5.3 million Americans are living with a TBI-related disability, so a permanent disability from TBI. This is roughly the same amount of Americans living with Alzheimer's disease. Now, Alzheimer's is going to go up as a public health problem because our population is aging, but TBI is always there in the background. 25% of our combat casualties over the last decade have been to brain or spinal cord neurotrauma, and 250,000 people injured in the last 12 years in our military. So it's a major health problem for our military. And the other really big reason why I decided to focus on traumatic brain injury is that 30 clinical trials, and a phase 3 clinical trial means the drug or the cell or whatever that product was has already passed a safety trial of phase 1, a little efficacy trial, does the stuff work in phase 2, and then the phase 3 is the big, how does it work in a large blinded trial. 30 phase 3 clinical trials for TBI, they've all failed in the last 20 years. So we have no effective treatment for TBI. So what is a TBI? It's really quite simple to define. It's just an alteration in brain function or other evidence of brain pathology. Is the brain tissue damaged following force or a blow to the head? So it has to be an external force. If you have an internal injury like a stroke, that would not be considered a TBI. And then a lot of people ask, well, what's a concussion? And so a concussion is a subtype of traumatic brain injury. So it's just a minor or a mild form of TBI. And if you picture the brain inside a skull here, and you bump that skull, you can see the brain jostle around inside there. Now what's missing from this picture is you also have a fluid-filled space protecting the inside of your skull or the inside of your brain from the inside of your skull. So this is a little bit of a dramatic example, but it shows that the brain can jostle around in there and get bruised in the skull case. Now, because we're at a university, I'm required to have a pop quiz. So everybody take out a piece of paper. How many of you have had or think you've had a concussion? Wow, now I think this is a biased audience, probably because they saw what the title was. The answer, if you survey 3,000 Americans last year, is that 1 in 4 people have suffered a concussion. And this is pretty even across age groups, across socioeconomic status. 25% of Americans have had at least one concussion. And I didn't survey, but how many people have had multiple concussions? We don't know the answer to that, but a lot of us have had multiple concussions. I certainly have had multiple concussions, and I'm worried about what it's done to my brain. Now, moving to The primary causes of traumatic brain injury, 35% of them are from falls. So this is either in children learning to walk, they fall, bump their head. The elderly who have balance problems, they fall and bump their head. Another large problem is motor vehicle accidents and people getting struck by objects, hit by a baseball, using their iPhone and walking into a light pole. Believe me, it happens. So TBI can happen through a variety of causes. And the trouble is, where the brain gets injured alters what the problem the patient's going to have. So TBI can be a very global injury or it can be a very focal injury. So it varies across patients. And how to assess the patient on a uniform scale is not easy. So what we use is called the Glasgow Coma Scale. It ranges from a terrible score of 3, meaning you're completely unconscious, to a perfect score of 15. So if you're in the 13 to 15 range and you've not had a loss of consciousness, or if you've only had a loss of consciousness for 30 minutes, you'd be classified as a mild traumatic brain injury patient. And the symptoms typically subside over a couple of days, but not always. You can have permanent injury even from a mild TBI. But your symptoms of headaches, dizziness, concentration problems, those are typical of a mild TBI. And the more severe TBIs, you have loss of consciousness for between 30 minutes to a day. That would be a moderate TBI, or from more than a day is a severe TBI. So you have headaches that get worse or they don't actually go away. You have concentration problems, memory loss, problems speaking and understanding, and maybe even convulsions or seizures. So TBI is a very broad thing, but the way we measure it in patients is not very precise. So this adds to some of the difficulty. I don't know why that jumped. But the other thing I am very interested in is not just a single brain injury, but repeated injuries. And so there's a PBS special that you can download online or watch online called League of Denial. The NFL first helped support this documentary, and then later they pulled out and didn't want it shown. It's a fantastic documentary on what happens to football players who've had repeat mild concussions over the course of their career. Bennett Omalu was the first pathologist to identify chronic traumatic encephalopathy, this disease of individuals that had repeat injuries to the brain, repeat mild injuries. And so I highly recommend that you watch the movie Concussion as well. And Anne McKee has followed up on that work. What does it look like in the brain of a patient with a brain injury? So the top panel here, these little brown spots are the brain's immune cells. They're called microglia. And if we look at uninjured or control patients, we see that in young, middle-aged, and old people, the amount of inflammatory cells in the brain gradually increases, but not a lot. These are young people who've had a traumatic brain injury and they died within 1 month of that injury, and they have a lot more inflammation in their brains. If you look many years later at patients who've died subsequent to a TBI, that inflammation is there at 8 months, 4 years, up to 16 years post-mortem. So that inflammation is very severe. Also, though, we talk about white matter atrophy. What is that? So white matter are the nerve fibers that go from one hemisphere of the brain to the other. Very long fibers that are myelinated or insulated, so electrical signals can get to each side. And there's a marked atrophy over time with traumatic brain injury in people. And this is the first autopsy case of an NFL football player. This was Mike Webster of the Pittsburgh Steelers. And he was in Omalu's original report in 2005. So it's 13 years later. This pathology kind of looked a little bit like Alzheimer's disease. But following up on Omalu's work, Anne McKee has done an autopsy now on 111 NFL football players who've donated their brains, and 110 of the 111 had this type of pathology. So this is an early stage bit of pathology. This is a low power and then a high magnification. Anything in brown is a pathological structure in the brain tissue. Stage 1, stage 2, stage 3, and stage 4. This looks like almost looks like full-blown Alzheimer's disease, essentially, but it's in the brains of somebody who's 30 or 40 years old. We also know that we can see this pathology in college and even high school football players who've suffered concussions. So Anne McKee went on and classified the scale and the stage. Anything in dark orange here shows more and more pathology, and this pathology is rather extreme. Can we model this in an animal and learn more about what's happening? All right, so that was your background. and we're now moving on to human neural stem cells to treat traumatic brain injuries. So this is work being done in my laboratory and funded by CIRM, the agency that Dr. Anderson was telling you about. Then I'll talk about my modeling of repeat mild concussions using brain organoids or stem cells in a dish to model TBI, and then we'll move on to the SAN-BIO trial. So, the model that we typically use is called a controlled cortical impact. And a controlled cortical impact is a model where you anesthetize a rat or a mouse, you trephine or drill a hole in one side of their skull. So you don't damage the brain, you only remove the bone. Then they're positioned in a device that holds their head steady, and a pneumatic piston is positioned over the center of that open skull. And then a computer controls a piston to cause a force blow to the brain tissue. Now what this allows us to do is to give every rat in a study the exact same injury. So it's very artificial. It's very effective. I can dial the injury into a certain amount and get the animals to have a certain amount of memory impairment, certain amount of motor disorders. It's very unlike the human population where their injuries are all over the map. But this is the most common model of a traumatic brain injury in a rodent. And as of a year or so ago, there were about 50 publications looking at a human stem cell of some sort to treat traumatic brain injury in a rodent. So this table, which you can't read, but you'll see it online if you want to, this table shows you basically that a whole bunch of people have looked at a whole bunch of different kinds of cells. The bottom line is very few people report whether their cells even survive the transplant in the animal model. That's kind of a problem as far as I'm concerned. And very few studies have shown that there's any behavioral recovery. So as of 2016, only 6 of the studies were long-term, meaning over 2 months in duration, giving the stem cells enough time to differentiate and turn into something in the animal host. Very few report cell survival, and only 4 of those 50 publications show that there were improvements in memory. So there's not a lot out there on the horizon. And so we decided to turn to a neural stem cell and test if neural stem cells could treat this problem in rodents. So a little bit of basic science here. Sperm meets egg. It forms a morula that divides into a blastocyst. This blastocyst is what you would get from an in vitro fertilization clinic. So if somebody is discarding embryos from an in vitro fertilization clinic, if you take the inner cell mass, you can get embryonic stem cells from this IVF clinic medical waste. So colleagues of mine in Sheffield, England have done this for several cell lines, and I'm going to show you that data in a moment. You might get stem cells from aborted human fetuses. You might get stem cells from different organs of the body. The SAN-BIO trial I'm going to tell you about harvests bone marrow and gets the mesenchymal stem cells from bone marrow. And there's a new technology that we're also using here at UCI, which is to take skin or tissue from an organ and induce them to become pluripotent stem cells. So those are called iPS cells, and they're from an adult donor. So there's no fetus or there's no embryo or no embryonic stem cells involved. So there's lots of sources of stem cells. The question is, which ones work, why do they work, and can we find ones that work better? So about 5 years ago, I started collaborating with Harry Moore at Sheffield University in England. He sent me a cell line called Chef6, and we took Chef6 and we had to transition it from cell culture media, so stuff that the cells grow in a dish, and that stuff had animal products in it. And you don't want to transplant something into a person that's been exposed to animal products. So we had to transition these cells from an animal-based media to chemically defined media that doesn't have contaminants in it. So we came up with a process to do that, and at the end, after about 3 months in culture, we've generated a neural stem cell line that is capable of differentiating into cells of the nervous system. So that's the summary of Chef6 development. So I'm going to call them Chef6 human neural stem cells. That's what HNSC stands for. And our study is going to be, let's have some sham animals that aren't injured at all, but our technicians don't know who's who. A group of animals that will be injured, but they get a vehicle that the stem cells are delivered in, but they don't get any stem cells. Some of the animals get stem cells that have been in culture for many months, and some of them get stem cells that have been in culture for just a few months. And then we randomize all of these animals, and we look at what happens. So this is a whole brain from a rat in that study, and this is 5 months after the injury. You can actually see a cavity, and that cavity is shown here. So one hemisphere is severely damaged. If we look at that in 3D, we can reconstruct the size of that lesion and where the lesion is, and we can see that the lesion is very consistent across the animals in our study. We can also go in and look for the human cells that we transplanted. And so every little dot you see here represents about 4,000 human cells that have survived and where they migrated on the side of the brain we transplanted them to. So we can figure out how well the cells engraft into the host and how large their damage is. Now we want to see, do these cells do anything? Do the animals improve? So one of the examples we use is something called a novel place recognition test. This test is showing a rat two objects in one location, and after they've learned these two locations, the next day you put them back in the box and you move one of the objects. I call this the who moved my cheese. So what this looks like, this is 4 separate boxes, and we're going to put the rats in the box, and they are looking to see where are these objects, and they're exploring those objects, and a computer is tracking where they go. We can actually generate heat maps that show us where did animal 1, 2, 3, and 4 go. We get a lot of numerical data out of this saying what did the animals do. But the important thing is to say if you're a TBI injured animal, you do not discriminate your negative scores for what got moved. You can't remember what got moved. But if you've received the Chef6 neural stem cells, you almost return to normal levels. So that dotted green line is what an uninjured animal would do. So we see that these human neural stem cells actually improve the place recognition in this task, and the rats are slightly better. Another thing we can do is we can ask them how well do they remember a hidden platform. So this is a water maze, and it's a cloudy milk-colored water. There's a hidden platform here, and this rat is swimming around trying to find that platform. On day 1, he hasn't a clue where the platform is. So he's got to swim around, and eventually he's going to find that platform. If he doesn't find it after 4 minutes, we actually show him where it is. We don't want him to drown. He's a test subject. We care about him. But eventually, hopefully, he'll find that platform. Now, this is the same guy 5 days later. Boink. He knows where to go. So we can also plot this with heat maps and say, where did this guy swim to? And here's where he's gone after 5 days. And we find that if we give brain-injured animals human neural stem cells, they are able to learn this task again. Whereas if you brain damage them, they look like this. They can never learn that task. So what do the cells look like when we put them into the brain? This is a slice of rat tissue, and in red is a marker for all of the human cells. So these are all the human cells that survived this transplant, and in green is a marker for what's called NUN, or immature neurons. And we can see that a lot of these human cells turned into neurons. They turn into the glial cells, the supporting cells that myelinate and nurture that tissue. And we also found something interesting, and that is if you injure an animal and you count the host cells, so how many rat neurons are left in the hippocampus, they have a severe loss of neurons if you give them an injury and just the vehicle. Baseline, or where an uninjured animal should be, is this green dotted line. If we transplant the neural stem cells, we return them back to almost to baseline. So they're preserving the neurons of the host. They're also turning into new neurons and new glial cells to support that host. So that was part 1, a summary that Chef6 human neural stem cells improve performance on these learning tasks, that they protect the host, but they also turn into new neural tissue themselves, that we want to figure out now, can we increase the survival of these cells and extend the time to transplant? So right now we've done 9-day delay between injury and transplant, and we want to extend that longer. Can we treat a patient who was injured a year ago instead of a patient who was injured just 2 weeks ago? Extend our time window. And so right now we are funded by CIRM to test what are called GMP cells, or good manufacturing practices cells. That means the Chef6 cells that we received have been treated in a way that is compatible with FDA guidelines and regulations to put them into people. So we're replicating what I just showed you, but we're doing it with a new batch of Chef6 cells that would be allowed to be used in people. So that's the next bit, and 2 papers have been published in the last 2 years. You can look that up online if you'd like to. So I've finished with that part. We'll move on to repeat mild concussions. So repeat mild concussions, they're a major problem in the military. Our troops exposed to IEDs and getting concussions, rocket launchers, grenades. People don't think about it, but a troop being trained how to use a rocket launcher or a mortar is being exposed to concussions repeatedly. Also, our kids are playing water polo and getting hit in the head with the ball. They're doing martial arts, playing soccer, heading the ball, and of course playing football. So there's a lot of things that could lead to repeat mild brain injuries. So we wanted to model this in an animal. So our design is as follows. We take an animal and we anesthetize it. It's laying on a foam pad. This is the same metal piston that we would hit the brain with, only this time we don't open up their skull and the animal is not fixed in place. So when they get hit, they actually just bounce and they recover very quickly. So it's a very mild form of injury. And the design is give an animal one injury and look at what their behavior is like. Give an animal 5 injuries over 5 days, or 5 injuries over 5 days, or 5 injuries over 10 days, or give an animal 10 injuries 10 days in a row. And then we want to ask, are there behavioral differences in these different animals? What we see here is a test called a rotor rod. So I don't have a video of this, but this is a rotating rod, and a rat or a mouse can walk across that rod, and as it rotates, they move and they can stay balanced on that rod. But as time goes on, the rod speeds up. So they fall off the rod, and this is how long does it take them to fall off the rod. When they're uninjured, they can do this pretty well, and over time, they even learn this task better. If they're injured, they're impaired, but only on the first day or two, and then they recover as well. So this concussion model is actually a very, very mild injury because it doesn't result in permanent motor deficits. However, if we look at their emotional behavior, how do they perform in an elevated plus maze? So a rat or a mouse should stay hidden in the closed arms of a maze, and if there's an open arm, they shouldn't go in there. But if they're injured repeatedly, they spend more time in the open arms than the animals that have been uninjured. So there's something, they're more risk tolerant. They're more aggressive. If I show you this water maze task, it's the same task. They're supposed to learn. So these two, the pink and the blue, are the sham animals who've never been injured, or the animals that received one concussion. And they learn this task over time perfectly fine. They get faster and faster and faster. The 5-hit animals and the 10-hit animals, they never learn this task, even though one concussion is incredibly mild. If you look 6 months later, so they were injured 6 months ago, and then you test them on the water maze, they can't learn this task. And that's from a few repeat mild concussions. What does the brain look like? If we do a cross-section of the brain and I stain it for the insulating protein that surrounds the fiber tracts or the axons, we see that a sham animal has a very thick corpus callosum, we call this. This is the superhighway between left and right hemisphere. Sham animal, uninjured, very thick corpus callosum, same with an animal with a single concussion. But even non-experts could see there's something wrong with the corpus callosum compared to the sham animal if they've received 5 or 10 hits. Now we can actually model this in 3D, and this is just the corpus callosum, or the white matter connecting left and right hemispheres. And we see a very thick corpus callosum here and a thinning over the 10-hit animals. We can actually then measure the volume of their tissue, and we see shams and 1-hits have no tissue loss, whereas the 5 and 10-hit animals have a tissue loss, just like that corpus callosum loss I showed you in the humans with traumatic brain injury. We can also look at their inflammatory cells after the injury, and we can see that there's a lot of inflammatory cells that are still present in that tissue months later. So now we're using this model with colleagues here to study whether the genes that are linked to Alzheimer's disease, if you give a concussion to these animals, will they develop Alzheimer's pathology faster? And also if we can model chronic traumatic encephalopathy in these animals. And I'm sorry, let me go back. We're looking at the relationship between the inflammatory cell and how animals get migraine and pain with my colleague Kathy Cahill. We're looking at whether we can block this inflammatory activation and prevent this pathology from happening. So is there a therapeutic approach to this? Or now that we know there's permanent damage, would stem cell therapy also work in this animal model? And finally, we're looking for metabolomics or biomarkers of concussion. Can you look at the blood from an injured animal and detect that they've had a concussion, and can we develop this into a sideline test for injury in either troops or in football players. And that's with my colleagues Massimo Fendaca and Howard Fedoroff here at UCI. And lastly, we're going to move on to 3D brain organoids. I call them Borganoids because I'm a Star Trek fan. And we can grow these human cells into brain tissue in a dish, and then we can injure the human tissue in a dish. We can kind of skip the mouse or the rat altogether. So I did not invent this. This was invented by a woman named Madeline Lancaster in Austria a couple of years ago. But this is images from my laboratory where you take embryonic stem cells, you transform them and put them into little matrigel, they're little jello-like droplets of nutrients, and you put these into a bioreactor and they can grow to very large sizes. So this is a bioreactor, a large bioreactor, and this is a 12-well plate that a CIRM intern, funded by CIRM, Mike Warren in my lab, has built this little device that I can put brain organoids into each of these different wells so I can keep track of them separately. So now we can grow little pieces of human brain tissue from embryonic stem cells. They can grow to be 3 or 4 millimeters in size. We can drop them into a micro 3D printed skull case so that we match the size to the whole of the brain organoid. And then we can injure these. If we use a laser system, a very gentle laser pulse, we can give them a mild concussion. If we use the injury device that I do the spinal cord or the brain injuries with, we can give them a severe brain injury. So what does this look like? This is a paper clip to give you an idea of the scale of a 35 millimeter dish. So this is a cell culture dish. We put media and reagents in there to feed them. We drop our fabricated skull case in there, and then we gently put a brain organoid into the most closely matching skull size. We put this dish onto the platform of a laser microscope, and we generate a little tsunami, a little micro pulse. So this is, that was a mistake. Let me fast forward here. This is what it looks like in the microscope. This is the brain organoid here. You can't see all of it because it's very big. That's where the laser is focused. And each time we pulse the laser, it creates a shock wave and it concusses that brain organoid and bounces it off the wall of that skull that we fabricated. So what happens to this tissue? If we don't injure the tissue, we don't see anything except this blue marker of cells. But the green marker is a marker of early Alzheimer's pathology. And in a concussed brain organoid, we actually see the coup, or where the laser was focused, injury, and the contra coup side, where it bounced off the wall. We have mild injury on both sides. If you zoom in on this, you can see individual cells, and the green is this hyperphosphorylated tau protein that is a very early marker of Alzheimer's pathology. So we're actually seeing that we can model and generate Alzheimer's in a dish. So that concludes the brain organoid bit. I've told you about my human neural stem cells and how they can improve recovery from brain injury. I've told you about the repeat mild injury model in rodents, and then growing little human brain facsimiles in a dish and injuring them. I want to thank my team who's involved in this. It's been a lot of people, funding from a lot of different agencies, but really without these people, I wouldn't be able to do any of this. So I can't name them all by name, we don't have time, but thank you, thank you. Very talented bunch of people. And I need to move on to what mesenchymal stem cells are. So to make it very clear, the first part of that was my research here in the Stem Cell Center. Now, because SanBio is not here today, I'm just going to tell you about their mesenchymal stem cells. So this is not my data, but SanBio is a company that has taken from bone marrow, they have harvested bone marrow from volunteers, they grow those up in a flask, and then they expand them. And then they transfect them, or they infect them with a virus carrying a gene that will make them produce supporting factors that will help tissue heal. So that's their hypothesis. Once they do that, they can cryopreserve these or grow them up in massive banks and store them in freezers, and the product is called SanBio 623. So this is a engineered mesenchymal stem cell. Now if you take SB 623 and you put it in a dish, you can measure the cytokines or the inflammatory cascade markers that these cells produce. And basically what they're showing you is that in comparison to a mesenchymal stem cell that has not been transfected with this gene, SB623 makes more of most of these trophic factors and most of these cytokines. So they've made a cell population that they think is going to help preserve brain tissue. If you plate rat neurons or nerve tissue in a dish and then you give them media that's been conditioned by normal mesenchymal stem cells or SanBio 623, the 623 generates more neurons, more astrocytes, and more glia. So they're showing that these cells promote growth of neural tissue in a dish. They also have an assay called the aortic ring assay. You put tissue down and then put media in with this dish, and you look for how the capillaries grow out into the center of that ring. And the more capillary growth, the more your product supports new growth of blood vessels. And so they find that the SB623 improves branching of arteries. They don't last a long time when you transplant them. They're not tumorigenic, so they don't form tumors in animals or in people. And they've also published that they demonstrate improvement in a rat model of stroke. So lastly, they recently published that in a brain injury model, these are rodents with a brain injury very similar to the model that I just showed you, that the animals are improved at 1, 2, and 3 months on an elevated swing test so that their balance is improved. Their rotorod task that I told you about, the rotorod task is improved compared to vehicle 1, 2, and 3 months after the transplant. And their overall neurological score, how good is their balance and how good is their ability to walk on a narrow beam, those are all improved. So that is the background of why SanBio wanted to go to a clinical trial with mesenchymal stem cells for traumatic brain injury. And now that's the end of my section. I'm turning you over to Dr. Chen, do you have a call with a patient right now? We had to switch mics. And Dr. Chen is going to update us on SanBio and how you get these cells into people. Next.
All right, thank you, Dr. Cummings. So I'm going to talk a bit about some of the clinical implications of the stem cell trial and a little bit about head injury as we see it as neurosurgeons. So I'm a neurosurgeon up at the medical center. All right. And so there are challenges that we face, all right, in getting these stem cells into the patients. So I'm going to talk a little bit sort of about the nuts and bolts of how we do it and what are the challenges we face and our experience a bit that we've had with this first stem cell trial. Right. No disclosures. Right. And first of all, as Dr. Cummings had mentioned, there's a whole spectrum with traumatic brain injury. The most common, where there are probably about 3 million or so concussions per year in the United States, but there are also probably about 100,000 severe traumatic brain injuries in the US, all right? So there's a spectrum of it. So not all head injuries are the same. And because of that, I think when you direct your therapy, when you're directing your stem cells, you have to keep that in mind, all right? Some of these we treat with surgery. Some of these are treated with medical measures, all right, where we give medications to decrease the swelling of the brain, decrease the edema, try to protect the cells. And some are treated with both. Some we can't treat at all, okay? But there are many different factors that affect the outcome, right? And just to show you that not all traumatic brain injuries are the same, here's an example of a patient who's 78-year-old who fell back, hit the back of his head, and you have this contra-crew injury here, okay? And a few hours later, he comes in, initially he's talking and responsive, interactive, is put in the ICU. Our residents watch him, keep an eye on him, but we notice that over time, he deteriorates over time, and you can see that there's this blossoming or increase both in the brain injury here, this contra-crew injury, as well as this subdural hematoma, all right? Let's contrast that to another patient, okay? Here's a 26-year-old male who was visiting our country from Colombia as a student, and this patient came in initially, again, Glasgow Coma score of 11. He has this bit of subarachnoid hemorrhage, a little bit of blood there. And we see that over time, that evolves, and he has a very similar picture that the other patient had, okay? And you can see that there's all these spots in the brain, which is blood, or hemorrhage, or damage to the brain. So the end result can be the same. You end up with both patients who have a low Glasgow Coma score and severe traumatic brain injury, and many of these patients undergo surgery. Some of them undergo medical treatment. All right. Here is an example of a patient who had a head injury and had a hemorrhage there, right? And what I like to point out is that over time, if we look at this patient now 6 months later, you see that there's this area of damage, this area of encephalomalacia. Okay, so there's been damage specifically to that, but if you recall the blood clot, it isn't quite as big as what we see here. There's a bigger area here. So what that says to us is it's not just the area that was damaged, but the surrounding area, or the penumbra area, is also damaged. All right? So what we do in neurosurgery and in our ICUs is we try to protect the brain, or you minimize the damage. So the whole talk, all the talk from the trauma team and trauma services, we're part of the level 1 trauma center, is avoiding secondary injury, avoiding secondary insults, avoiding hypotension or hypoxia. I mean, so that you want to make sure the brain gets enough blood and oxygen. That's what you want to do, all right? And so that way you can minimize the damage to the surrounding area, okay? Because you know there's an area that's damaged in the epicenter, but what about the area around that? So can you minimize that? Can you decrease that? All right. So some of the key points that I hope to illustrate with just a couple examples here is that traumatic brain injury is multifaceted, all right? And also traumatic brain injury evolves with time. So patients can show up at time 0, but over time that head injury progresses. There's more swelling, there's more edema, there's more bleeding, all right? So there's more damage. So it is not static. And many times this injury is not just during those golden hours, but it is over the subsequent days, over the next day, 2 days, 3 days, 4 days, even weeks, all right? And so, and also, the nature of the injury is also dependent upon the mechanisms of injury, all right? Dr. Cummings talked about there's different types of injuries, ground level falls, motor vehicle accidents, sports injuries, all these impart the kinetic energy in different manners and different ways. That's why traumatic brain injury is such a heterogeneous topic. And that's why when we talk about it, some of the studies that are done, when you try to design these clinical trials, why they are so difficult to also oftentimes achieve that statistical significance, because you're dealing with baseline patients that are very different, all right? Okay. The impact of the traumatic brain is dependent upon the location. So frontal lobe versus temporal lobe, right side versus left side. Patients who have left-sided injury frequently will have trouble with speech and language, all right? Right side may have more difficulty with abstract things, okay? But speech and language is oftentimes what we see most prominently and what affects the patient and the communications with family and patients most profoundly. So location is important. Age is a factor, okay? And particularly the extremes of age. Young patients, we know, tend to have more plasticity, all right? So they can hopefully regenerate a lot. Older patients do not. However, we are going through a phase where we have a plethora of older patients. In fact, the most common injury that we now see up at UC Irvine, head injury, is older patients who have a ground level fall, all right? And you think that a ground level fall means just basically where I am falling forward, okay? That isn't that much energy, that, you know, we all do it now, we all do it when we're young, we get by with it, okay? But what we're finding is that we have a large population, and this is nationwide, that people are getting older, all right? They have comorbidities, they have other medical problems, and they're falling, okay? They hit their heads, and oftentimes this can be devastating. Certainly, it can be life-changing. And that is in particular, we don't have a lot of information, a lot of good data as to how to treat that, and that is obviously something that we're studying up at the medical center. Okay. The long-term sequela, okay? So, we had talked about how not all head injuries are the same, right? But also, not all ICUs are the same, not all trauma centers are the same, not all neurotrauma centers are the same, and often what we do in the ICU, how we limit the injury, how we limit the spread of the secondary insult is going to affect the outcome. And this becomes important for stem cell trials because if we create a better environment for potential recovery, then the patients will have a better chance of having those stem cells work, okay? And so the area of damage, for example, if you make the area of damage smaller, if you make the area, the penumbra area, or that area which is at risk smaller, which has a potential to recover, then perhaps those stem cells will be more effective. So that's why I think that particularly we're very intent on this up at UC Irvine, is being very intent on brain monitoring, doing everything we can to minimize the secondary insult. Now, traditionally, secondary insult is, we talked about low blood pressure and low oxygenation, okay, but it's more than that, all right? So this is a quad Lubin bolt that our chief resident drew some pictures for me, okay, and this is an FDA-approved device where you can put in 4 different monitors in regionally the same area, okay? So you're checking brain pressure, which pretty much everybody does. You check brain oxygenation, okay, which probably about 50% of places are doing. But we check brain temperature, brain blood flow, and brain metabolism using what we call microdialysis, all right? And so you're sampling a small area of brain. So granted, it's a small area of brain. It's not all the brain, but it's a representative portion of the brain, all right? And if you're really aggressive, you can put one on each side. These are all FDA-approved devices. So none of this is research. This is our sort of our standard care for severely head-injured patients up at the medical center. And what we're trying to do is create the best environment for the brain to recover. So studies have shown if you optimize their glucose, you optimize their metabolism, all right, there's less cell death. There's less of that penumbra area. So that is just briefly some of the things we're doing to try to optimize our care of our patients. Okay, so you take care of the patient as well as you can and treat the head injury. Now you're left with the sequela of head injury. And as Dr. Cummings had mentioned, you have a large number of patients who are dealing with the sequela of head injury, all right? And these are patients who need help, whether it be family or caregivers or being in neuro rehab centers, all right, or in nursing facilities. And this is where the hope that we're looking for with these stem cell trials, all right? And Dr. Cummings talked about some of them. We were engaged in a randomized controlled trial up at UC Irvine of the STEMTRA trial, okay? And that is the mesenchymal stem cells that Dr. Cummings talked about. So the question is, how do you get those into the brain, okay? So there are barriers that are there naturally, and these barriers, include the skin, the bone, the dura. So all these linings of the brain you have to get through to get the stem cells into the brain. So these are natural barriers, and also the targets are often deep down in the brain, oftentimes several centimeters down in the brain, okay? So how do you get there, and how do you get there safely? Finally, you want to avoid damage to some of the normal pathways, some of the normal cells that you have to go through to get there, the normal layers, okay? Also, for patients who have had surgery, who have had craniotomies where the bone flap is taken off and put back on, there is the challenge of the previous surgery because you have to get through this area here, all right, which is oftentimes synthetic and harder to get through. Also, there is the risk of scarring underneath here, okay? The brain underneath may not be normal, it may be scarred, so getting through that is a challenge also. So these are some of the challenges that we think through sort of from how to do this, okay? Now, how do you get down there, okay? One of the methodologies that has been around for now many years is using stereotactic neurosurgery. So what that is basically securing a reference frame to the patient's head, and it creates coordinates which are then integrated into your X-ray, okay? So you get your X, Y, and Z based on the X-ray. In newer times, we're now using CAT scans and MRI scans and computer programs to help guide us to pick a spot, and using your XYZ coordinates and the azimuth, you can plan and target this to place your target or your stem cells to a particular area, to a particular depth, to a particular area in the brain. So this is rather laborious, and you have to sit there and spend a lot of time coordinating it. But that is the way that the stem cell trial was designed, okay? And this is the traditional method that has been used for this trial. Basically, the patient is put into a frame, and it has the coordinates there, all right? Then the patient goes to the operating room, all right? The patient goes to the operating room, and in this case, the patient's awake, so that is, they're not intubated, so they're breathing on their own, but they're sedated. It's like when you go to the dentist, you're sort of zoned out, but if we really push you and wake you up, you'll wake up and wiggle your fingers, all right? And part of that by design that they want the patients not to have general anesthesia. And so the patient then has the frame put on the head, you use a target to sort of guide you to drill a burr hole. And so you're trying to do all this through a burr hole, and this just shows the setup that is done. Okay, the setup, you make your cut, and then there's the tube going down into the brain. So here at UC Irvine, we were fortunate that we had a robot, okay, which is called the ROSA robot. And basically, what this is a robotic arm that will point us and direct us to our targets. So it does not do the surgery for you. What it does is it holds the arm in place. But what you can do is you go here to your planning station, and you plan where you're going to go, and you say, I want to go to this specific spot, and the robot will point to it and say, you need to go 10 centimeters deep, 14 centimeters deep, all right? And you can place your target right there, okay? And this navigation system is very nice because what it does, it uses the facial features, so it traces the facial features and integrates that into the computer and acts like a GPS system to direct us to this. Okay. And this is just to show there seems to be a cluster of them here, right in our area, the closest one being at Chalk Hospital, but they're spread out throughout the US, and I think that this is technology that's going to pick up because it does make it much easier for the patient and also for the surgeons to do the surgery because it streamlines the whole process. This just is some examples. Intraoperatively, you can look on the screen and plan your trajectory. It points you right to where you want to go. You drill your hole, and you then target or put your catheter or your cannula or your sheath down into the specific area where you want to go. Okay, again, some more pictures showing us drilling the hole and lining things up. Okay, so just again about this trial, okay, just a little bit about the trial. It's a multi-center, placebo, randomized controlled trial, all right, and it was done all over the country in about 25 centers, okay, and those centers included the screening centers as well as the surgical centers. And what happens is the way the trial is designed is that patients would go to a screening center. And so I'm not involved with that. That is a center where they have neurologists who will look at the patient and specifically look at the criteria, all right? They will say, okay, this patient has a motor deficit, so you're looking for a motor problem. So this trial is focusing on motor function. Okay, so the patient would have to have had a head injury, and the head injury would have been greater than 12 months ago. And why is that significant? Why is the 12 months significant? Because we know that patients can get better, okay, up to 12 months. So they get a lot of improvement up through the first 6 months to 12 months. So you want to maximize their improvement naturally first, because you don't want to bias the trial. So if you started them at 3 months and started putting stem cells in, how do you know it wasn't their natural progression that got them better, all right? And they had a fixed motor deficit, and then it has to be surgically accessible, all right? So that is where we as neurosurgeons have to look at it and say, well, you know, I think I can get to it. I think I can get to safely without a lot of collateral damage. And a lot of that has to do with looking at the frame. How's the frame put? If it's way back here, it's hard to get to, okay? But what we find with the ROSA, the robot, is there are more degrees of freedom, and we were able to get to areas that other people said, you know, couldn't be done, okay? And so I think that's where new technology, and UC Irvine has been great about investing in new technology to help, you know, not necessarily help us out, but it was there, okay? And it worked out very nicely. Okay, so the patients are randomized to receive placebo or 2 different concentrations of the cells. Okay, they have a 1 in 3 chance of getting the placebo, all right, which nobody wants, but that is part of that study, okay? And you have 15 injections along 3 tracks via single burr hole. So a little bit about clinical trials, okay, and why do you have to do it as a trial? Why can't you just these things work, okay? There's been one trial with stroke where it worked, and that's been published, came from Stanford and big places like Emory, so it's got to be good, right? Well, the issue is that the rigor, you need to have the rigor for a clinical trial, all right? And that's why you do it as a randomized trial, okay? And again, although everybody, their loved one, wants to get the stuff, okay, how do we know that the stuff really works? How do we know that there aren't problems with it? How do we know it doesn't cause tumors, okay? All right, so that's why when you do a trial, there's, a really good trial, there is a arm where it is a placebo or a sham arm, all right? And luckily, this is only 1/3 of them, all right? But that's sort of the importance of the trial, and why is it randomized? Why don't all the people who get stuff come to UC Irvine, right? No, we can't do that. We have to randomize across the country, all right? And then the importance of follow-up and the importance of blinding, okay? So when I put the cells in, I know what's there, but the neurologists or the screening people do not know, what I did, okay? And that's why we do a placebo, which is a sham, which is basically drill a hole, but we don't put the needles in the head, okay? So the patient thinks they have a cut there. Everybody sees this cut. They see a little hole there, so they believe they have it, because we know there's a placebo effect too. There is a placebo effect with all trials, and also if I were to evaluate the patient, I see them afterward. I think they all got great. They're all doing great, right? That's why we have to have an unbiased team, which is the screening team, who see these patients and follow them long term up to a year later, and they have rigorous documentation of how well does that arm move. Is it still, weak? Okay, they have metrics they follow, and they follow these patients and grade them. And so there are people who do that, whether it be the neurologist, whether it be their physical therapist or occupational therapist. You need to have that objective data. Okay, so that's the importance of follow-up. All right. Okay, so this again shows what you try to do: a single burr hole, okay, and 3 trajectories. Okay, so you're going to go 3 different trajectories, and you're going to put 15 points along here. Actually, 5 points along each one. So 15 total injections or little plugs of stem cells. Where do you put them? All right. So what you're trying to do is you're trying to put them near the area of injury. Okay. So I showed you previous CAT scan where we see the area of encephalomalacia. So you say, well, why don't you just put it in there, let it populate and refill that area? Well, the thought is if that's already all dead tissue there, okay, you don't have anything to reinvigorate or regenerate. So what we try to do is you look for an area that is near the area of injury. So here is the area of injury right here, which you can see better on this special scan here. Okay, so this area is not dead. This area is dead. So you don't want to put it in a dead area. You want to put it sort of around this area with the thought that perhaps these cells that are there in the peri-injury area are not quite dead. Maybe they're suppressed by inflammatory cells. Maybe they're idling. Maybe they're undergoing, a slow apoptosis or cell death. Okay, maybe they're still there. So what you're trying to do is invigorate those cells, and those mesenchymal stem cells that Dr. Cummings showed have the ability to give all those growth factors. So you try to put it near that rather than in the area that's dead, because you put it in the area that's dead, there's nothing to invigorate. Also, with these particular mesenchymal stem cells, they're only there for about a month. Okay, after a month, they've done studies, they're gone. Okay, so what's going on here? Well, they don't really know. Okay, they believe that somehow it activates something, okay, or suppresses the inflammatory response, or somehow activates the cells around there, okay, to get them going. How do they know they're gone? Well, these cells, these patients do not get immunosuppressants. So they do not get immunosuppressants, they do not get Decrejon, they do not get steroids, okay? So basically, they're going to get rejected, they're going to die off, all right, and they've been shown that it's after a month. But the studies have shown that they do something, okay? And actually what they do, we don't know. Area of intense debate, a lot of controversy, a lot of studies. Okay. And this is just another picture of that, okay? Again, showing to target that area. So our experience, we participated in this from February 2017 to the present, all right? And there are 2 trials here, okay? I have here, this is the SandBio stroke trial that we participated in. I think that there's another talk about that later on in the series. And this is the head injury trial here, okay? Both trials are now done. They've reached enrollment. They've completed their enrollment, okay? 61 patients here and 163 here. We put in 18 patients here and 6 patients here. We did these all with the ROSA, and we had patients coming from all over the country to have this done. It did very well, no complications. Whether or not it helped, again, I defer that to the centers that will be doing the screening. So this is just shows sort of the experience that it can be done safely. Getting those in there, initially looks difficult, and there's clinical or technical challenges that you have to overcome, but it can be done, and it can be done safely. So I think that is one hurdle that we've shown you can overcome. Now the question is, what do you put in there, and how do you get them to proliferate and do their thing? So how will we find out whether this works? Okay, so this stuff is currently being analyzed, but they will follow these patients out to about 12 months before they will have all their data. So they're going to keep looking at their data, analyzing it, looking for statistical significance, and hopefully, somewhere down in the springtime, we may have some data that will be presented that shows whether or not these work. I think whether or not it works, I think that there's a lot of promise in it, but as we know, there's been a lot of phase 3 trials that have failed, but we will learn from it. So I will stop there and have Dr. Anderson come back.
Stick around, leave it on.
So I'm just going to bring us on home here for a couple of minutes before everyone flees. But I really hope and invite you all to ask questions. We're going to have open questions. Anyone who's interested in anything about stem cells, hopefully we've given you a sense of this bench to bedside principle, translational research with stem cells, why it's very important to do these controlled clinical trials and understand the basic science, and maybe pique your curiosity a little bit. In that vein, I just want to show you a couple of slides about upcoming events. This community lecture series, this was our kickoff, will continue. Many of you may have received a flyer, a schedule of these events. There's also some again at the back of the room. In the fall, November 14th is our big Stem Cell Awareness Day symposium. We do a scientific symposium during the day. But last year we did our first large community lecture series. We had Elena Cattaneo from Italy, who's a senator for life, a tremendous investigator in stem cell research, who spoke last year. This year we're going to have Dr. Irving Weissman from Stanford, who is really the father of stem cell research, and tells an amazing story about where stem cells started and literally the identification of the first stem cells and understanding how they work and that they're there, and in his case, what role they play in cancer and how we might think about controlling them. So I invite you all to keep an eye out for that. And then lastly, I just want to end on this slide because again, a big part of our purpose here is really education and getting people to think about what they can do and how important, I hope, and that you can see stem cell research is, and being involved as community members. And so here's just a list of things that we get asked about what you can do. Number one is to participate. And so Americans for Cures is here tonight. Dr. Mitra Hushmand is in the audience, for example. This is a grassroots organization that drove forward the first of the California Institute for Regenerative Medicine Prop 71 bills. We'd be delighted to have you participate in that. Again, there's information at the back of the room. Contact your representatives about whether it's stem cells or not, science research, and that this is something that is important, and you can see that there's the potential to really be transformative in terms of what our potential is for treating all kinds of things, not just traumatic brain injury down the road. And then last of all, we have a newsletter, we have Facebook, we encourage you to sign up, and of course we have a very active philanthropy program supporting our seed grants and other initiatives. You can find that on our website, or we have some representatives here tonight. So with that, thanks very much, and I'd like to turn on the lights and open up for questions.
Something I found very interesting, and you see this clinically, and I guess it's hard to see it translated necessarily, not only in animal models, but also in humans. But for example, physiologically, we see that children do very well after traumatic brain injury. They have significant recovery as compared to, older or elderly adults. And in part due, we believe, to neuroplasticity, the ability of these neurons to make new connections. So it was very interesting that myelin, the myelin slide, looking at the brain sections, looking at the corpus callosum in the animal models. I think it'd be very interesting to see if these, these Chef cells, for example, these neural stem cells, are able to make the corpus callosum more robust, or create, make new pathways for connections, or like induce more plasticity. Obviously, we've seen that they're able to, help with behavioral changes in animals that are close to non-injured rodents. And same, I guess, in humans, it'd be interesting to look at the axonal regeneration or ability to make new connections in some of these patients that we've implanted these stem cells in. Do you think that's something we'll look into?
So that's actually next on the list.
Okay.
So we didn't expect that these very mild concussions were going to have this severe of a physical change in the brain. So, being a neurosurgeon, you understand that, but to the public, one concussion is a very mild bruise. You recover from that just fine. But if you have repeated concussions, there's an additive event there. And we were shocked to see how severe it was. And these were in young adult animals. So now, next on the list is, can we were not expecting that stem cells would actually be a therapy for repeat mild concussions. We thought the acute model, severe head injury, motor vehicle accident, that type of thing, was what you'd treat. You'd use stem cells to treat. But certainly on the horizon, we're going to want to test the stem cells in the repeat mild concussion model.
Maybe just to your point briefly, and I don't know, Jeff, can you
unmic so that
hear
we
us
can
back there?
pass
That around if we need to.
Okay, yes.
I don't see where the mic up here. So part of the question, right, is how can it be that young creatures, humans included, have so much more plasticity, so much more recovery than might be the case in older individuals? And one of the reasons we see this in animal models as well is that the brain is still developing and growing, and there's a very active stem cell population in all of the neurogenic regions of the brain, and that starts to decline with aging, and it declines much earlier than you might think. So adult in a rodent is around 3 months of age, and already those stem cell populations are starting to drop off. And in humans, that's happening, unfortunately for all of us, in your teens and certainly in your 20s. And so, and it declines continuously throughout life. And so our innate capacity for repair is not what it is when we're kids. So a different question, right, at the very basic science end of the spectrum is, can we wake those cells up, right? So in adults, can we wake those cells up, and now maybe in the long run not have to transplant cells, but be able to activate our own stem cells and kind of heal thyself sort of theme?
Go ahead.
When you inject the stem cells into the brain, clear the damaged brain tissue, how do those stem cells know what to do? Or do they act on surrounding cells, or are they like in charge now and they tell everybody else what to do?
So, the question, as I understand it, is we put the stem cells in there. How do they, know what to activate, what to connect to, what to try to invigorate? Is that correct? Okay. So I think that, as Dr. Cummings showed, these stem cells have the ability to secrete or make lots of different growth factors. So we don't know exactly which growth factor is the one that's working. But I think, again, that the stem cells will go in there and they will implant themselves, and they will secrete whatever they're made to secrete. And that is what's going to activate the cells around there. So it's not specifically like going to a specific tract or a specific area. What it does is it somehow activates the cells that are there. And so we don't really know. And I think a lot of this, if you look at some of the original papers, and that they still don't know which cells are being activated, but they create a better environment. So they're saying they're secreting growth factors that can activate, cells that are not quite dead yet.
I would add in the spinal cord injury model that Dr. Anderson talked about very early on, we did find that when we transplanted those human cells into an injured spinal cord, they actually made new insulation and wrapped the axons of the mouse. So those cells, because they differentiated or turned into a newly born oligodendrocyte, we call that's a myelinating cell, we saw those cells wrap the axons of the injured mouse. And we also saw that the human cells could turn into new neurons, and those neurons would actually make a chemical connection with the mouse neurons. And we know that, which was a very cool experiment, we repaired the spinal cords of these mice, restored their ability to walk, and then we gave the mice diphtheria toxin, which is lethal to people and harmless to rodents. So we killed all the human cells that had been transplanted into that animal, and the animal returned back to unable to walk again as if it had never been transplanted. So we know that the cells integrated or connected up with the host, the injured mouse, but that's a neural stem cell. That's not the mesenchymal stem cell. So as Dr. Chen was saying, we know that the mesenchymal stem cells, they only last a month or less in the tissue, and then they die off. So they've repaired as much as they're going to repair in that first 30 days, and then that treatment's over. Sir?
Yeah, right now you're putting the stem cells around the damaged part, and you're talking about you don't want to put them inside because of the lack of, or damage inside, lack of blood vessels and so forth. Do they have new technologies coming in, like hydrogel and so forth, scaffolding they can possibly put in there to survive? These cells can survive, so possibly you can get more improvement?
Yes, so the question is, do we avoid the epicenter, or the middle part of the most damaged tissue? But could you actually transplant cells into a matrix or some gel that would hold them there and help support them while they matured and differentiated into the cell type they're supposed to become? And we actually do work with biomaterials, scaffolds that can hold the cells, or polymer gels that can hold the cells as well. That's part of a big ongoing research program, and particularly Dr. Anderson and I are working with a colleague, Lonnie Shea, at Michigan, who's making these biomaterials. We also have faculty here at UCI, Lisa Flanagan, will be talking in maybe 6 months about her bioengineering efforts as well. So that work is ongoing. Dr. Hushman of America for Cures, a plant.
Well, beautiful series of talks, went beyond expectations as always. But two questions for you. One, can you comment on your plans on the organoids and whether or not you intend to test different treatments? Is there a way to use drugs to see the effect of those drugs on these organoids? And secondly, sort of a philosophical question, do these organoids have feelings or consciousness of any kind?
Great questions about the borganoids, or the brain organoids in a dish. So let's tackle the philosophical one first. Do they think? Are they philosophically alive? My pat answer is no. They don't have a nervous system. They don't have input from eyes and ears and sensory organs to even know that they exist. They're not a fully mature brain. They're just a facsimile of a brain. So they're a good model to test what human brain tissue does. If you look at them with a little electrode, they do signal each other, so there's a network of communication going on that Lancaster showed is feasible. But in terms of are they thinking, no, they're far too primitive for that. What might we do with them? So I had to give a very short talk, and I think I went over as it was. So I'd be happy to stay here and answer a lot more questions. Yes, the whole plan of those borganoids is you take one stem cell from a person, and you can make 20 clones, if you will, of a brain facsimile that are all very, very similar. So the genetic code is the same. And now I can, using that 12-well plate, I could take 12 separate brain organoids, and I could injure 6 and not injure 6. And then I could let them mature after the injury. And of those 6, I could give drugs, 6 different drugs to those, and compare them to that drug given to the uninjured ones. And I could screen for therapeutic agents much faster than using a giant flask, and much less expensive as well. So that is exactly the idea, is can we screen for biomarkers of injury in these brain organoids, and then can we screen for drugs that might prevent injury or restore normal tissue? So not a plant, but a great question. Not a plant. Somebody else who I don't know. Sir.
So I got a few questions. So where's the best place for stem cells? And I mean that like the best place in the body, like bone marrow, fat tissue, cord blood, embryonic, autologous. I know it's tough, but I also wanted to ask, like, what places in the world are really doing great stem cell research too? I mean, it's not just.
You have to ask us.
Yeah, I'm. So the question, we'll go backwards because that's how my brain works. What's the best place in the world for stem cells? I'm slightly biased, but not much. I'd say not the United States, but California specifically. CIRM has done an incredible job of stealing stem cell biologists from all over the world and bringing them to California. That was actually the plan. Let's fund stem cell research so specifically that scientists will want to come here to do their stem cell research here. And that's worked very well. So we have an alpha stem cell clinic that you heard, network that you've heard about, that nobody else has that. So California really is ahead of the game, although it's competitive. There's, Japan, Korea, Europe are also doing very good stem cell research. Australia is as well. It's all over the world, but California is really an epicenter for it. And the second question you had was, what stem cell's the best? So of course the answer is Chef 6. You knew that before you even asked it. But that's not actually true. My response back to you is, well, for what problem? And so we really don't know what's the best stem cell for a brain injury. Is that the same stem cells for a stroke? So Dr. Chen has transplanted SB623 into stroke and TBI. Both of them have a inflammatory problem, and both of them could use that support that those cells provide. But that's not what you're going to want to give somebody with Parkinson's disease, and probably not for somebody with Alzheimer's disease. So I think it really depends on what the thing, fat tissue and stem cells from fat, they're not going to treat brain injury, but they might treat cartilage damage or shoulder injuries or something. So it really depends on what injury you're thinking about. And I can tell,
I want to sneak in on this
and
question.
being married for more than 3 years to this woman, take both of those.
Take both of those. All right, that's just complicated. So what I want to chime in on is two things, right? So one part embedded in your question and that I'm not sure everybody necessarily gets is this idea of autologous, right, versus allograft. And so autologous means your cells back into your own body. And an allograft is like an organ transplant, right, from a donor to a recipient. And we match those theoretically as well as we can. And that's what's going on with most stem cell trials right now. So the huge advantage potentially of autologous, so that would be to make an induced pluripotent or a reprogrammed stem cell from your fibroblasts, from your fat cells, from what have you, and then differentiate that, bring it up the branches of the tree so that then you can transplant it for what ails you, stroke, for TBI, for spinal cord injury, diabetes, whatever. And the huge potential advantage is that your body would hopefully see it as self and not reject that cell, right? So as Jefferson mentioned, in reality, mesenchymal cells don't like being in the central nervous system very much. Those are the SAN-bio cells. And on top of that, they're allografts. And so your body, the people who receive these cells are starting to reject them over time. And so they don't stick around a long time. We're counting on them doing nice things, making a friendlier environment in order to have their effect. But if you really want them to integrate, like other neural stem cells that Brian and I have worked with, then you have to have them not be rejected. And so that means you either have to do self into self, or we need a much better way to control the immune system than we have right now. Both of those are actually moving along pretty fast in terms of research. I honestly think we're going to get to similar places with those two strategies. The catch is, coming back to the basic science principle, is we still can't control super well how to convince a pluripotent cell, one that's at the base of that tree, to be something in particular and sort of go all in, be committed to it. And so the trouble that we have with induced pluripotent cells or fat cells that have been transformed, this kind of thing, is we can't convince them in a dish very well to be, for example, a neuron in the case of traumatic brain injury. And so if we can't do that, either they're not going to do very well in terms of repairing by integrating, or we have a pesky problem with those in their potential to make tumors. And so that's where our basic science comes in, right? Because we're still in relatively early days of trying to learn how to control those cells in a dish, tell them what to do and where to go after we transplant them so that we can make the most of them therapeutically. Does that make sense? Yeah. Oh, come on, it's a very friendly crowd here. There you go.
I'm an amateur. I have a son who's 40 years old with a traumatic brain injury. And I've been to some seminars on stem cells and what they can do. A lot of companies are promoting them. And one of the things that they have been saying is that the umbilical cord stem cells are kind of like virgins, and that they won't be rejected because that's what they're engineered for, going from the mother to the child. Is that false?
Yes. But let's repeat the question, which is, you have a 40-year-old son who has a brain injury, and you're hearing from clinics that umbilical cord blood or stem cells from umbilical cord, they won't be rejected because they're virginal, and so they're a good source of cells for transplantation. And this is a topic for a whole nother hour lecture, but basically people believed that the brain was immunoprivileged, meaning the immune system couldn't get into the brain. So if you put something foreign in there, it wouldn't get rejected. Well, that's actually wrong. And then the second thing people thought was that embryonic stem cells or umbilical stem cells, they don't have any antigens or markers on their surface that would tell the host that there's a foreign cell there. Wrong, not true, and they get rejected. And so, in fact, it was recently that we thought mesenchymal stem cells, they won't get rejected because they're an anti-immune, anti-inflammatory cell type. And so if you transplant them, they'll stick around because they're dampening the immune system. Not true, they get rejected. So the rejection problem is there. It's the solution to that is probably going to be generating stem cells from your own skin, but we're not there yet in an industrial sense. And I can also tell you're wanting to chime in. So, but first, I will ask if Dr. Chen has anything to add to that.
Go.
No, not really. I think you summarized it nicely.
Okay, then chime in.
So I would just come back, and because it's an excellent question, and there are, between Los Angeles and San Diego, there are at least 40 clinics that are offering mostly either autologous cells or things like umbilical cord cells, which are a kind of mesenchymal cell, for kind of everything in the kitchen sink, right? Same cell, diabetes, arthritis, ALS, Huntington's disease, Parkinson's disease, Alzheimer's disease, autism, right? There's kind of no delicate way to say it, right? In classic snake oil fashion, that's what you would expect, right? If there's something that's a cure-all, you should look at that with some suspicion from the get-go. So the real question is, what are you asking those cells to do, right? Because as Dr. Chen told you, the cells that get implanted don't necessarily have to survive long-term. If they're just making a better environment, or if they're changing the immune response to make a friendlier place for your own brain or your own kidney or your own pancreas to function, that might be enough depending upon what we're expecting them to do. So I wouldn't say that those are bad cells per se. It really depends what kind of disease, what kind of injury you're talking about, where you're talking about putting those cells. And then again, back to that basic biology question, what are we asking them to do? What's the mechanism? And that's where everything has to integrate through that bench to bedside. And so the trouble with most of the umbilical cord stem cell research that's out there, or autologous fat cells, for example, is that it's a lot of hope about what they might do, but not much on the basic science and practical mechanism, understanding what they're really doing. Does that make sense?
And I will chime in, because that made me think about it, is that the question, and Dr. Chen really explained this well, I felt, was that you want to be in a controlled clinical trial, an FDA-approved clinical trial. You want to be in a clinical trial in the alpha stem cell clinics that are funded by CIRM, because you're part of an actual scientific experiment that's been ethically evaluated, the protocols have been approved, and it's very easy for little fly-by-night companies to come up with a product, sell you on some hope, and they don't really have any data behind it. People, for example, in Florida have gone blind because they were treated with some harmless cell that in fact wasn't harmless. So I would do your research, check our website, because we have links to things like you should be going to clinicaltrials.gov, the International Society for Stem Cell Research.
My son was almost in the clinical trial, but he had received Botox for his spasticity.
Yes.
So he wasn't able to participate because you wouldn't know if it was the Botox or
Right,
the stem cell
right.
network.
So that's another good example, is if somebody goes and gets an umbilical cord transplant, then they're going to be excluded from the next neural stem cell trial because they're not a fresh patient. So it's complicated.
These are very difficult.
I gotta say something. Dr. Chen is dying now to say something.
So, just wanted to add that, with this trial, okay, just because you didn't get in doesn't mean that there won't be other trials. And I think there's a lot of hope with this trial because they did do their 50% analysis. So with all these clinical trials, they got to get to 50%. They do an interim analysis. And if it is shown that it is not efficacious at all or it harms patients, it gets canceled. So that it did get to the 50% mark so that they went and completed the whole trial. So there is hope to this trial. And I think that, whether this trial or other trials, I think it's very important that, it's a whole team that participates, okay? Because anything with the brain, you have to get into the brain, okay? And at this point in time, the only people who can get in the brain is neurosurgeons. And as we saw, there are challenges to it, and there are pitfalls to it, and you can cause a lot of harm or damage if you don't do it right.
Sir.
Oh, I'm sorry. Do you mind if we go here, just because you haven't had a chance yet?
Talking about the ethics of it, I was just wondering, what is the gauge of the needles that you're sticking in the brain? What kind of damage do they do to the brain? Already sticking them into a already damaged area. And I certainly wouldn't want my loved one to be one of the 30% in the placebo arm.
Right. So the question is, okay, just repeating it for the other audience, is the gauge of the needle, okay, that you're putting, the cannula that you're putting in, number one, and number two, what about the ethics of, 1/3 of the people not getting the stuff, okay? So with respect to the needle, that is probably about 1.5 millimeters in diameter. So 1.5 to 2 millimeters in diameter. So that is equivalent to the standard ones that we use for doing biopsies. And so there's been a lot of data that shows that what happens when you put that in, the tissue just parts, okay, even if you're going through normal brain. So those are standard cannulas that we use for biopsies. So we use those routinely for biopsies going deep into the brain. And the risk of a bleed or a hemorrhage is extremely low from that, okay, probably less than 1%. And we frequently put in even larger catheters and 5 millimeter catheters without any problem. So, and again, that's where with our ROSA system, it makes it easier because you can look and plan where all the sulci are, where the vessels are, so you avoid those. Right. Now, with respect to the...
It's magic.
So with respect to, the ethics of, 1 in 3, okay, not getting the stuff and going through a sham surgery, that is something, we explain to the family and patients way up front, okay, so they know that, okay, and, they can choose not to do that, and that is really their choice, and is really explained up front that, for the rigor of the science, okay, and most patients are fine with it. Their families, because if they don't get the stuff, they're at least contributing to science. They're contributing to overall helping people because the knowledge that you get from that okay, is important. Also, because it's a clinical trial, how do you know that the 2/3 who are getting this stuff aren't going to have a not good outcome? So we don't know that. So it goes both ways too. So I think that a lot of people are fixated on just like getting the stuff, but we have to be circumspect and realize that the stuff may not do what it's supposed to do, okay? Or there may be some unforeseen, bad outcome or bad result that we can't predict. So they're also taking a risk. The people who are actually getting the stuff too. So I think it goes both ways.
Do you want to mention about crossover trials?
So this one did not have a crossover trial, but sometimes there are trials where patients may start along one pathway, but when you do your 50% evaluation, or it's been clearly shown that there is benefit, they will cross over the other way. Okay, so that wasn't built in this one because it's such a small trial, but many trials, a bigger trial, and I think what you have to look out for is if this trial is statistically successful or shows that there is a benefit, okay, then there'll be a phase 3 trial, okay, where it'll be even more rigorous, where there will be a crossover. So again, a lot of that is sort of by design. That is sort of the rigor before the FDA is going to put their stamp on and say this is the way we should treat all head injuries. Okay, that is the rigor, and that sort of is the beauty of the U.S. that there is this rigor that we as physicians are doing something that has gone through the test of a clinical trial. I mean, in other countries, is not quite as rigorous.
Yeah, and the last thing we would want to do is stumble into making somebody worse, right? So testing, being a well-informed clinical trial where that's really being tested is the goal. It's an experiment at that stage. Sorry. This gentleman's been waiting a long time. Go.
Those Chef cells you were talking about, are they the same as Reneuron is using over in the UK, CD34? Is that the same thing as Neuron?
That is a great question. And so I'm actually, I believe, so don't quote me on this and turn off the recording devices. I think they are using a Chef embryonic stem cell line, but it's not Chef 6. So the part I can tell you for certain is it's not Chef 6 because I have an agreement with the University of Sheffield for neurotrauma with Chef 6. But I think that, I know that Sheffield has 12 or 15 ES lines that they have doled out to various investigators. So it might actually be one of those. Ma'am.
Yes, with regard to these cells, we know that the clinicians are involved in this and also companies. And let's face it, this is all for profit. So what is the intellectual property involvement with these companies that are offering you these cells? Is there any type of potential conflict?
So that's the question is everything. So everything is for profit. What's the conflict of interest with having a company sponsoring a trial or sponsoring your research, or who owns the product, who owns the discovery? So those are very related questions, and it really depends on what the trial is. So first, I would say mostly in an academic setting, we are a nonprofit. So we get a paycheck from federal grants or state grants that help pay our salary. But if I discover something, it's owned in part by CIRM, who funded it, and it's owned by the university. And I'm a socialist. I'm here at the university to discover stuff. So a lot of us are just trying to do good and discover things. And the complication is I could be as socialist as I'd like, and if I discover that Chef 6 does something, it needs to go the next step. So I'm not going to make a profit from that, and I'm fine with that. But the negative side would be if no one picks it up, then we've done all this research, and the state of California has invested in Chef 6 as a possible therapeutic, and no company picks it up to go further unless they can have part of the intellectual property of that. So that's beyond my pay grade, but what happens is, so were I to make the next step and show that the new manufactured cells work, I would hopefully be getting a grant from CIRM again to manufacture them. And in that process, I would be licensing that technology to somebody to manufacture them, and then getting more funding to do a clinical trial. I wouldn't be the one, I wouldn't be paying for that. I also wouldn't be making the profit from that.
Forgive me, I didn't mean to imply.
Oh, that's quite okay.
Any type of profit involved here, but that was my issue, I guess I didn't put it properly, is the intellectual property issue of these cells.
So the intellectual property issue is it's too bad Lester left. So we had an intellectual property lawyer in the audience here tonight who's supported us before and has actually lectured in our series. So intellectual property is owned by the university, the institution where the discoveries are made. And so in the case of SanBio's cells, though, SanBio owns those cells, they manufactured them, and they're contracting with the university to do a trial. So we're happy to help, we want to help, but if that becomes a product for treating traumatic brain injury, that's going to be SanBio's product, and they're going to make the profit from that.
And so again, just to do my thing and expound slightly, it's complicated, right? So you have a sense of that from Brian. Our entire basic science research system in the US, National Institutes of Health, from the way it was founded, is built around the idea of discovery and moving things forward and having somebody else pick up the development cost. So we're in the States, very capitalistic society, and that's what it's built around. So part of that has to do with cost, because basic science investment is relatively small on the scale. Even CIRM, who has made an enormous effort to bring things through, right? I talked about the 49 trials that they've invested in to bring things that are stem cell based or stem cell product based or stem cells themselves through to clinical trial. Their investment is in the phase 1, phase 2 stage. So they're putting in something on the order of $20 million per trial for a starter trial. To do a phase 3 actual multi-center randomized controlled trial, now you're into $100 million or more, and that moves the threshold completely into the realm of having companies do that. So at that point, what we can hope to do is make a discovery, convince somebody that it's interesting to do an actual trial in the clinical setting, and then the entire system is absolutely dependent upon that looking promising enough for a company to pick it up and run with it. NIH is investing, similar to CIRM, a little bit in doing those foundational trials. UCI just got funding to be part of a NeuroNext network, which is a clinical trial based very much like the Alpha Stem Cell Clinic from the National Institutes of Health to be able to drive these responsible, well-controlled trials forward. But again, those ones that are in the very early stage to show proof of concept, to get over the threshold just from a cost perspective and what it costs to develop the cells and do all of the assessments, enroll all of the subjects that you need to do a randomized multi-site trial is out of the spectrum of anything that's a nonprofit.
All right. I think we're gonna break for now, and you can come up and ask individual questions. We'll stay, but that's you're dismissed. Class is dismissed. You're free to go home. Thank you so much.